Hello and welcome everyone to this video podcast for evolution and diversity. In the last video, we talked about the rise of eukaryotes. And in that video, I also introduced you to the protist. And in this video, we're going to look more closely at a protist. More specifically, we're going to talk about the many types of adaptations protists have and how that has allowed them to be evolutionarily successful. So when you see in this picture here, these are all examples of protists. And so you can see there's a wide variety of types of protists out there. We'll see single-cell protists, multi-cell protists. We'll see protists that have the ability to move in different ways, a whole variety of them. There is a huge variation within this lineage of eukaryotes, the protists. So what we're going to talk about today are these adaptations that we find in protists. We're going to talk about variations we see with the nucleus, or when there's more than one nucleus, the nuclei. We're going to talk a little bit about support and protection adaptations, cell numbers, going from single cell to clusters of cells to multicellular, the different adaptations that protists have acquired to help in feeding, that is acquiring energy, adaptations to help in motility, and adaptations for reproduction. All of these have led to the wide variety of protists that we see. But let's go ahead and get started with the discussion of the nucleus and nuclei in protists. All right, so let's focus on some of the variations that we see with the nucleus or nuclei that we find in protists. Now, most protists will have a single nucleus per cell. However, there are some examples of protists that have multiple nuclei. And here are just three examples. First, we see here in the Giardia a case where we see two nuclei. In fact, they are identical nuclei. Okay, another example of having multiple nuclei is seen with the paramecium. Paramecium have two nuclei, but they're not identical. The macronucleus is larger and appears to have more of an important role in daily functions. Whereas the micronucleus is smaller, as you might imagine with a name like micronucleus. And you can see the micronucleus here, and it is much smaller, and the macronucleus, which is much larger. But they're usually right next to each other. And this is the germline nucleus. This is what gets passed on to the next generation. So when progeny are made from this paramecium, the micronucleus is the one that's passed on to the next cell, and it will ultimately give rise to the macronucleus. Now, as the last example of looking at protists with different numbers of nuclei is the slime mold here. And it's rather spectacular looking, and it's hard to see here, but this is a single cell. But it contains millions of nuclei. So while most protists contain a single nucleus, we see here are three different examples of either containing identical pairs of nuclei that have different sizes and different functions, and in this extreme case, a single cell with millions of nuclei in them. Next, let's talk about the different kinds of support and protection that different protists have. Now, maybe one of my favorite are seen here with this type of protist called the diatoms. And now this is a very interesting protist in the sense that its cell wall is essentially made of glass. It's a silicon matrix, but it's essentially very similar to glass. This middle one here is a dinoflagellate. And its outer cell wall here is made of cellulose. Cellulose is a type of a carbohydrate that is very long and very extensive and contains many connected glucose molecules. Another example includes the foraminifera. And it doesn't have a glass wall or a cellulose wall, but rather it has a wall that is made of calcium carbonate. It's much like a shell. And the last example of a support and protection adaptation is seen in Euglena, which you see a picture of it here. Now, this is the only one that we're talking about where the adaptation is on the inside. And it forms something that we call a pellicle. And the pellicle is essentially strips of proteins. And mainly what we're talking about here are microtubules that are part of the cytoskeleton. Now, protists can also vary in the number of cells that make up an organism. They can either be unicellular, like we see here in these diatoms that we talked about on the last whiteboard. They can also be colonial cells, Each one of these cells performs a similar life function, but they're not unicellular because they live together within this larger structure shown here. So we say they are colonial. And an example of colonial cells are seen with the Volvox, which this is a picture of here on the bottom. One of my favorites. And then finally, protists can also exist as multicellular organisms, like we see here in this giant kelp, in this large kelp forest. So these represent a multicellular organism. And unlike we see with the colonial cells in the Volvox, here we see cell specialization. And the cells are dependent on each other. Let's move on to this next adaptation that protists have. And these adaptations we're going to talk about are going to be associated with how protists have adapted to feeding and obtaining nutrients. Many protists are autotrophs. And remember, autotrophs are organisms that can make their own organic nutrients. They are capable of undergoing photosynthesis. Now, there are many types of protists that can undergo photosynthesis. Some of these include some algae, euglena, and diatoms. Now, in addition, many protists are actually heterotrophs. And heterotrophs obtain their organic nutrients from other organisms. Within protists, I want to talk about two different ways that they can obtain these nutrients from other organisms. The first will be absorptive. They're able to absorb those nutrients from the environment, much like bacteria and archaea would have done. The other way protists that are heterotrophs can obtain their nutrients is through ingestive mechanisms. Let's first talk about absorptive mechanisms. As I said before, many protists will obtain their nutrients through an absorptive mechanism. And this really just means that they absorb nutrients from the environment. These nutrients will pass through the plasma membrane and pass on into the cell to provide the nutrition that the cells need to survive. There are three different ways that protists can use absorption in order to survive. And I'm going to speak a little bit about each of them. The first is that protists can be decomposers. The second is that they can be osmotrophs. And the last one is that absorptive protists can be parasites. Let's begin with decomposers. So they obtain their nutrients from the environment by feeding off dead organic material. A better way to refer to dead organic material is to call it detritus. A really good example of these decomposers are the slime molds. I'm going to take a break from the whiteboard here and show a video of how these slime molds will decompose dead organic material. And then we'll come back to the whiteboard and move on to the other mechanisms of absorption. All right, let's next talk about osmotrophs. Osmotrophs feed by taking in dissolved organic material into the cell by extracellular digestive mechanisms. And the key to this definition for osmotrophs is to think about extracellular mechanisms. We're often familiar with intracellular digestive mechanisms when larger compounds are taken inside of the cell and they're broken down. With extracellular mechanisms, the individual organism secretes various enzymes that digests the material outside of the cell, and then it brings in that digested material, the organic material that is, into the cell. In a few moments, we're going to talk about the other way that heterotrophic protists obtain food, and that is through ingestion. And ingestion will involve intracellular mechanisms, because we want to finish off these absorptive mechanisms. And the last one here are the parasites. All right, so in talking about parasites, remember that some protists will live in other organisms. Sometimes that harms the other organism and sometimes it doesn't. When they do harm the other organism, they're called parasites. Some examples of parasitic protists include trypanosomes, and they're associated with causing sleeping sickness. Another one are the Giardia, which we talked about earlier. And remember, these cause something called beaver fever. The reason we call it beaver fever is because one of the first times that humans came down with this sickness, they got it from drinking water that was contaminated with Giardia that came from beavers. And the third example are some Plasmodium cause malaria. The last feeding adaptation I want to talk about are those associated with ingesting. And in talking about this, I want to describe two examples. And the first will be an example using pseudopodia. And I'll use an example of the amoeba. Pseudopodia breaks down to pseudo, meaning false or fake, and podia, meaning feet. And when we look at a video of this, you'll see how as the plasma membrane projects forward, moving, it looks kind of like the amoeba is stepping forward, if you use your imagination. But it's not a real foot, it's a fake foot. And then the other mechanism of ingesting that I want to talk about is ciliary sweeping currents. And an example of this that we'll talk about is the paramecium. This process of ingesting food, either by pseudopodia or these ciliary sweeping currents, is called phagocytosis. It's also important to remember that when we're talking about phagocytosis, these ingesting mechanisms here, that this involves intracellular digestion. So depending on the protist that we're talking about, they will either take in a whole living bacterium, archaea, a fungi, or another protist. And once they bring that into the cells, they will digest it using enzymes within the cell, intracellular. And when they do that, it breaks down those organisms into usable organic material. All right, I'd like to show a couple videos showing how phagocytosis occurs using these pseudopodia or by using these ciliary sweeping currents. So let's go to the video. Okay, so in this video, you see this very large amoeba here in the middle. And then you'll see this other protist moving towards it, or just accidentally bumping into it. But you can see how the amoeba is very slowly moving towards it. And this other protist doesn't quite know what's happening yet. Sometimes you look at this and you wonder, how is it possible that the amoeba is actually going to catch it because it's moving so slow? It kind of makes me think about The Walking Dead. You know, how is it possible that these zombies who move very slow can actually catch a human that can move fairly fast? But it's similar to this, right? This amoeba is moving so slow, yet this other protist, who is a little faster moving, doesn't quite know what's happening quite yet. And before long, it's going to get lodged in here and fall right into the amoeba's trap. Oh, it's too late now. Yeah, this little protist has swam its last day here. It's about to be consumed by this amoeba. Probably fairly embarrassing at this point since it's moving so slow, but this is what happens. And as the pseudopodia wrap around this other protist, as you see here, it will eventually encircle it and bring it into the cell. And once part of the cell in the cytoplasm, it will begin to undergo intracellular digestion. Okay, so here's another very interesting protist here that has this tail that's sweeping out, right? And it looks like it's fishing for some kind of meal. And in many ways it is. And you're going to see here in a moment, as it's extending out, it's eventually going to ensnare this other organism here. Oh, it's got it. Right now it's going to slowly wheel it in. And what happens here, it's hard to see, but right here, it begins to encircle it with the plasma membrane of this protist here, and brings it inside of the whole cell, just like the pseudopodia did on the amoeba. But this does a slightly different way by like sweeping out and catching it. But the end goal is going to be the same: that it brings this cell into this protist so that it can digest it. And here's just some more fishing expeditions going on here. All right, so we just looked at a couple examples of how pseudopodia can be used to bring in cells from the outside and then digest it within the cytoplasm of the cell, of the protist cell. What I want to do now is show you a video showing this ciliary sweeping current. And before I start the video, I want to show you a few features here. Right here, where you can see my cursor, and moving into here, that is called the cytostome. Essentially, that is the mouth of the cell mouth, cytostome. And then this structure here, this is a food vacuole. So food's going to come in through the cytostome. These could be bacterial cells or other small cells, and they will be filtered into this food vacuole. Once this food vacuole is full, it will pinch off, and then it will move on to become digested. And then this mouth doesn't quit eating. It just constantly eats. And so a new food vacuole will form. And we'll see that here in this video. It's a pretty good video, I think. So you can see this current, right, moving in here. What's harder to see is all the cilia along here that is sweeping food particles into this cytostome, the mouth. And you're about to see it pinch off this food vacuole because it's full. It can only get so big. It's about to pop off, I think. And there it goes. Now it's the mouth, like I said before, it doesn't stop. It's forming another food vacuole. Kind of fell out of the vision there. But it's going to keep filling these food vacuoles over and over again. Again, you couldn't see it very well, but those cilia all around the cytoplasm of the cell are constantly sweeping that fluid, that motion towards the cytostome. So the cytostome can constantly feed and eat all the particles that are coming into it. And then those food vacuoles fill up. Once they are full and they cannot take any more, they pinch off from the cytostome, and they then go on to digest the material within the food vacuole. Okay, let's go ahead and move to this next adaptations that we see in protists. And these are going to focus on motility and locomotion. I'm going to begin by looking at the amoeboid movement, named so after the amoeba here. And as I'll show in a video in just a moment, you can see that all these pseudopodia that we talked about before when we were thinking about feeding are used in moving this amoeba cell in a particular direction. The cytoplasm will extend and push against the plasma membrane, forcing these pseudopodia to move in that one direction. Now, a lot of the mechanisms that are involved in this movement are largely not understood, but it's gaining a lot of interest because some of the cells of our immune system move by this amoeboid movement. So by studying this less derived organism, the amoeba, we can learn more about the human immune system. Let's move to the video so you can see how these amoeba move in this amoeboid-like movement. So you see the amoeba here, and you can see it slowly moving to the upper right corner. And you can see the extension of the pseudopodia and how as it's moving, all the cellular components in the cytoplasm are also moving as well. Very slow movement, but it gets the job done. All right, that was the amoeboid movement. Now let's think about this next type of movement. And these are cilia. Now cilia, as you can see in this picture, completely surround the cell. On all surfaces of the plasma membrane, on the outside of the cell, you will see these cilia. And these will allow not only for the movement of food particles into the cytostome for feeding, but they also allow for the movement of these specific types of protists. Similarly, flagella. Now these are typically found as a single flagellum that help move this particular protist in one direction. All right, now I'd like to move to a video and show you both the cilia and the flagella and how they move. And here you see the cilia moving this protist, and you can see the cilia on the outside of the cell, and you can see it quickly beating so that it can move in that direction. likely to some sort of food source, but it is moving it. Here you see a euglena, and you can see the flagella on the back side of it here, on the left side. And it's moving it in a particular direction. And as it moves, you can see there is this rather impressive movement of the material in the cytoplasm as it moves in this one direction. Okay, let's now move on to our final adaptation that we're going to talk about with the protists, and that is reproduction. Okay, as we get started here, let's review a couple terms that'll help us as we move through this discussion. And the first terms I want to talk about are diploid versus haploid cells. Diploid cells we often refer to as 2N, meaning there are 2 copies of every chromosome. So for instance, in humans, all of our cells, except our sex cells, are diploid. All of our cells, except for those sex cells, have 2 copies of every one of our chromosomes. So in humans, just to give you a frame of reference, we have 2 copies of each of our 23 chromosomes, for a total of 46 chromosomes. Haploid cells we refer to as 1N, because it's just 1 copy of each chromosome. So to give you that same reference for humans, haploid cells are only our sex cells, are only our egg and sperm. All other cells are diploid. So in humans, our haploid cells have 1 copy of each of our 23 chromosomes. So in sperm and eggs, there are 23 chromosomes in humans. Now in protists and other eukaryotes, there would be different numbers of these chromosomes. Every species has a unique set of chromosomes. Next, I wanted to go over sexual versus asexual reproduction. So first, let's talk about sexual reproduction. One thing we need to remember is that sexual reproduction is uniquely eukaryotic. Only eukaryotes have sexual reproduction. In sexual reproduction, each parent produces a gamete. These gametes are haploid, 1N. Gametes are produced through a cell division process called meiosis. Essentially, meiosis takes a diploid cell, 2N, and produces haploid cells, or haploid gametes. It's also important to remember that in this process, we generate genetic diversity. When you take genetics, you'll go into much greater detail with meiosis. Gametes can be different, like sperm and egg, or they can be similar, like spores. Finally, gametes fuse to form a diploid offspring. All right, now let's talk about asexual reproduction. We see asexual reproduction in all domains of life. There are some differences, but essentially asexual reproduction can be seen in all domains of life. So this is any form of reproduction when the offspring inherit DNA from one parent. In protists and other eukaryotes, asexual reproduction is performed through mitosis. This can be either a diploid cell dividing and producing another diploid cell. It could also be a haploid cell dividing and producing a haploid cell. A key thing with mitosis is no genetic diversity is generated. So essentially we are making identical cells. You can get some mutations, so you can get some differences, but in general we think of this as producing identical cells. Now remember, protists can either be unicellular or multicellular, but they both still undergo reproductive cycles. With unicellular reproduction, the protist life cycle is dominated by either a haploid or diploid state. And I'm going to go to a PowerPoint slide in just a moment to walk you through this because I think it's a little easier to see using some of the figures from the book on this one. Multicellular protists have something that we call an alternation of generations. In this process, the multicellular organism will alternate between a sporophyte generation and a gametophyte generation. And I'll talk about that in a moment as well. But first, let's look at unicellular reproduction in protists. Okay, now I'd like to show you cell reproduction in unicellular protists. And I'm going to begin with talking about the situation where the protist is dominated by the haploid stage of life. In your textbook, this is found on figure 27.16. So here we have a haploid cell. Remember, because we see this one N here. This is the mature cell. This is the dominant form in this type of protist. It's going to undergo asexual reproduction, and it's going to use mitosis. And it's going to keep dividing over and over again, producing additional haploid cells. And it will continue to do this. Again, these are haploid cells dividing by mitosis. Then these haploid cells can interact with other gametes, other haploid cells, and they can fuse together by a process of fertilization. And then the two nuclei from each of the parents, the two haploid nuclei, now will also fuse together, producing a diploid cell. It will then undergo meiosis, and when it undergoes meiosis, it will go from this one diploid cell to haploid cells. And then once again, these haploid cells will continue to divide by mitosis here until such time comes where they will fuse with another gamete. But again, it will be dominated by the haploid cycle. This is the mature cell. Now, also in figure 27.16 in your book, we see what it's like if a protist is dominated by a diploid state. So now in this figure, I'm not going to walk it through piece by piece. I'm just going to refer to different parts here. So we have our diploid cell here. It will divide by mitosis, producing two new cells, and each of these cells will be diploid. Because that's what happens with mitosis. You go from a 2N cell to another 2N cell. It produces identical cells. Now, it will continue to do this over and over again. The resulting cell from a mitotic event gets smaller and smaller until it is too small to continue to divide by mitosis. At that point, this diploid cell will enter into a meiotic cell cycle, going from a 2N cell to a 1N cell, a haploid cell, either a sperm or an egg. They will then fuse with each other through fertilization, creating a diploid cell. Again, through meiosis, you go from a 2N cell to a 1N cell. These 1N cells will then fuse with each other through fertilization, producing a diploid cell. This diploid cell now is the mature state for this type of protist. And once again, it will continue to go through mitosis. And it will keep doing this cycle over and over again, producing a small enough cell that will then go through meiosis to produce these gametes. So again, this is the unicellular protist that is dominated by the diploid state. Now, as I said on the previous whiteboard, multicellular organisms undergo alternation of generation, so they're a little different. For instance, brown algae and red algae, these are both multicellular protists. Now, you can look at your figure in your book, 2717, and it's a good figure, and it goes through a lot of details, but I think what this figure here shows us is a much more simplified view of what's happening with the alternation of generations. So let me walk you through this figure here. You'll see that we have two different states here. One is the sporophyte, and one is the gametophyte. The sporophyte is always diploid, 2N, and the gametophyte is always haploid, 1N. The sporophyte here, being 2N, it will undergo meiosis. When it undergoes meiosis, it produces spores. And these spores, each one of these, are haploid, 1N. These spores then will undergo mitosis and produce this gametophyte stage. Each one of these spores in this gametophyte, they're all 1N. When they're released from the gametophyte here, they will form these gametes. And the gametes will then fuse with each other to produce a zygote. A zygote is just a one-cell embryo, but it's going to be 2N, diploid, because it's taking 1N from this cell and 1N from the other cell. Then the zygote, the 2N, will continuously divide by mitosis. And all the cells that it makes will make up this stage, the sporophyte. Again, each one of these cells in the sporophyte will be diploid, 2N. All of them will be. And then the cycle continues. These diploid cells in the sporophyte will undergo meiosis to produce the haploids. The haploids will grow through mitosis to produce the gametophyte. And then they will produce the gametes that will fuse to, once again, make the sporophyte stage. So it alternates between a gametophyte stage and a sporophyte stage. Okay, that is all I have for this video podcast for today's material. If you have any questions at all, please come see me or send me an email. And if not, I will see you in class. Bye for now.